Good afternoon, everyone, and welcome to my presentation. As you can see on the screen, my topic today is assessing genetic structure and reconstructing invasion routes of wheat midges on Canadian prairies. This talk is designed to give an idea about one particular part of my proposed PhD thesis, and my name is Kanishka, and I am currently under the supervision of Dr. Boyd Mori at the Department of Agriculture, Food and Nutrition Science of the University of Alberta. As we all know, prairies are popularly known as the breadbasket of Canada, as they provided the highest wheat production of the entire country. However, maintaining this threat is tricky due to the damage of numerous invasive species, especially invasive insects. Invasive insect species can have drastic consequences on the ecosystems, both in the natural and managed systems. In agriculture, invasive species can cause significant damage. So, is there any solution for this? Maybe what we are proposing today could be one solution for this. We can use population genetics as a tool to identify potential corridors of movements and population interconnections that can help to develop strategies to prevent the further invasion or the movements of these pests. This is one good example here, and this study has been done to understand the genetic structure and migration patterns of diamondback moths from the southern and to the northern region of China. This research provides a successful example of using population genetics approaches to resolve the seasonal migration of insects. Let's talk about villain of our story today. That is the wheat midges. Wheat midge is a significant pest of wheat cultivation. The recent years, significant wheat midge damage has been reported in Alberta, Saskatchewan, Manitoba, and some part of USA like Minnesota, North Dakota, and Idaho. Their long-range dispersal events through wind or other anthropogenic activities may have contribute to the current wheat meat distribution on the prairies. Why did I call it as a villain? It is because the damage that they cause. Wheat midge larvae feed on the developing wheat seed and trigger shriveling and pre-sprouting damages to the wheat cultivars. These damages will then facilitate fungal infections and eventually cause severe yield losses. Wheat midge damages are difficult to assess as the larvae attack from the inside of the wheat grain and as well as hidden within the ear. So they can do a huge damage without even noticing it. So to control them, it's important to understand their movements, blooming time, and other important life history traits. However, understanding the movements of the wheat midge is difficult as several different processes may influence their movement. For example, their movements are very short range and their movements are stepwise, but their movements are wind mediated. So wheat midge monitoring is currently relies on time consuming in-field counts on a daily basis during the susceptible crop stage. However, determining the movements of mobile insect pests like wheat midge is difficult just based on monitoring alone. So we proposed molecular methods as a tool for wheat midge monitoring. Understanding the genetic structure of different populations across their natural geographical range is vital to execute species-specific management plans. These two examples here shows the investigating the genetic diversity and population genetic structure of insects help in order to assess the planning of controlling programs. The one in the right side is about an invasive Mediterranean fruit fly in South Africa, and the one in the left is about a mosquito population in northern Italy. They have used what they have found through the population genetics to improve insect management plans more effectively than ever before. Molecular studies have been employed to infer insect movements across agricultural landscape and thereby predict corridors that are used for insect invasions. As we know, population genetic analysis can identify patterns of movements and potential invasion routes of these important pests. Also, investigating the genetic background and migration routes of source populations has important implications for determining appropriate chemical control measures because historical information on the insecticide treatments used on the different source populations at their site of origin and throughout their migration pathways could be vital elements to determine the most suitable control method for insect pests. Compared to other insect pests, there has been only a few population genetic studies have been conducted on wheat midges. Recently, there is a report about complete genome assembly of wheat midges as well. However, these studies are based on Chinese wheat midge populations. Recently, a North American-based study have conducted under the supervision of Dr. Boyd Maury, but this study is only based on mitochondrial genes. So up to date, a comprehensive investigation of worldwide wheat midge populations is still lacking. So considering all these informations, we developed following hypotheses and objectives. So we hypothesized that among population genetic variations in wheat midges will be structured according to the geographical distance, migration routes, and their source populations. Therefore, we will be able to trace their migration routes and trace them back to their original populations. Based on that, we came into an objective that to reconstruct the routes of invasions of wheat midges on the prairies and to determine the origin of the wheat midges in North America. So let's talk a little bit about how we're going to achieve this task. So we will collect wheat midge populations across North America and possibly Europe through different collaborators. 20 to 30 specimens of adult wheat midges will be collected from the wheat fields from different geographical populations. Once we have these samples, then we will subject these samples to DNA extractions, followed by DNA sequencing. Once we get this sequence data, then we can extract a single nucleotide polymorphism or the SNPs data out of these sequencing data. Then we can use those SNP data to analyze or assess the population genetic structure of these pests. Especially we can use DAPCs, admixture analysis, structure plot analysis to assess the population structure. Not only that, we will use outlier analysis and assessment of FST differentiations between coding and non-coding region of the genomes. So this outlier analysis will give idea about genomic locations or the markers that show behaviors or patterns of variations that are extremely divergent from the rest of the genome, as well as these FST values given a quantitative idea about how much these populations are different from each other. So once we have this data will then be used to compare the signatures of selection between invaded populations which are from North America and the populations which are from Europe, that are the native populations, and also the areas of recently introduced populations that will be Peace River region of Alberta, and populations which are currently established in central Alberta. So after that, we will be able to determine their possible invasion routes and their interconnections. The current management of these notorious pests are mostly based on applying insecticides. However, these insecticide practices are harmful to the environment, and the efficiency of these insecticides depend on the timing of control. Extreme use of insecticide against these insect pests can be cause the development of insecticide resistance and environmental contaminations. Therefore, this emphasizes the importance of developing, monitoring, and forecasting strategies that will help in the decision making during the insect pest control. With that, I would like to thank all of the funding agencies as well as the University of Alberta. And I would like to give away my thank you for my supervisor, Dr. Boyd Mori, and entire team. And thank you everyone for listening. And now I would like to take your questions and most importantly, your suggestions. Thank you so much.